So listen, everyone, thank you very much again for joining our today's lecture, our latest cyber series, where we have people at Mount Sinai present their own work. And today it's a great pleasure to introduce Dr. John Crary, who's a professor in neuropathology and leads Mount Sinai's Brain Collection, which is a major initiative to increase our own collection of human brains from individuals who pass away with a range of brain disorders. It's been wonderful for me to work with John over the years, and I'm looking forward to his talk today. John, take it away.
Eric, thanks so much for that introduction. As you know, I love working at Mount Sinai. There's so much amazing neuroscience going on here, and married with the clinical programs, it's just a formula for success. And so it's just been so enabling for me and for our neuropathology work. So let me see. And also thanks to Veronica for organizing and scheduling this conference. I'm going to talk today about tauopathies. And this is what I've been obsessed with and bent on for the past 15, actually probably longer, 21 years of my life. And I'm going to explain to you why I've become so interested in them. There are a whole bunch of neurodegenerative diseases that are associated with abnormal toxic aggregates of the tau protein in the brain. They, depending on depending on how you classify them, there are over 23 different brain diseases. They can have different symptoms. They have movement disorder or cognitive impairment. Some have motor neuron disease or psychiatric illness, and these symptoms can overlap. When you look at these patients' brains histologically, post-mortem, you see tau aggregates, not just in neurons, but in glia. You see oligodendrocytes and astrocytes accumulate tau. When you study the tau biochemically, you see different isoforms, different splicing forms of the tau protein, the different secondary modifications, different ultrastructures of the protein, and these also broadly overlap among the tauopathies, and there's different genetic risk among these different cases. There are mutations in the gene that encodes tau, the microtubule-associated protein tau gene. There are also common variants in the tau locus or genome-wide that also seem to confer risk for developing neurofibrillary tangles. To study the tauopathies, you really need to take an interdisciplinary approach. You have to work closely with clinicians, biochemists, geneticists, pathologists, psychologists, psychiatrists. And so we're really lucky to be at a large center like Mount Sinai, where all of these people are here. They're all interested in the same types of questions that I'm interested in. So that's really the formula for success. We know that there are different mechanisms. All of these mechanisms are not going on simultaneously in one patient, but we use these clinical features and what we know about histology, biochemistry, and genetics to kind of break these patients up into different categories. And the categories are kind of fluid, but the most common tauopathy, we consider it a secondary tauopathy. It's Alzheimer's disease. It's a monumental public health crisis. Rivals COVID perhaps in how important it is. There are currently over 5 million people in the United States living with Alzheimer's disease. It's very common. There are other common tauopathies. So the primary tauopathies are the tauopathies that are where tau aggregates are the main or the sole neuropathologic change that is evident in the brain. You may have heard of some of these like frontotemporal dementia, or chronic traumatic encephalopathy, which comes from exposure to repetitive concussions or traumatic brain injury. There are a lot of lesser known tauopathies that are kind of esoteric but important because they may have the potential to give clues to the mechanism of what causes tangles in Alzheimer's disease. There are certain rare brain tumors where you can get tangles, the ganglioglioma. It's what's interesting and potentially important in the context of neuro-COVID is that there are a couple of viruses that are known to cause neurofibrillary tangles. I'm not going to belabor this and go through all of them. What I'm going to do today is I'm going to focus on these age-related tauopathies, which has really consumed a great amount of my time over the years. So this slide is intended to give you an oversimplified overview of what causes neurofibrillary tangles to form. And if you just joined, please remember to mute yourself so that we don't have any background noise. Thank you. So the tau gene is encoded by a single gene on chromosome 17, MAPT, and it gives rise to multiple mRNA transcripts that are alternatively spliced that give rise to, in the brain, 6 predominant isoforms. These different tau isoforms have different functions, and they normally play a role in microtubule stability, axonal transport, neurite outgrowth, signal transduction. In certain contexts, tau becomes modified. These phosphate groups gets attached, gets hyperphosphorylated. This can be seen normally in just normal brain development and hibernation, but these aggregates do cause the tau to aggregate and form these oligomers, and then these oligomers can propagate from cell to cell. Then eventually they form the tangle, and that's thought to be what kills cells in neurodegenerative diseases. This may trigger neuroinflammation, oxidative stress, things like spread from cell to cell in a prion-like manner are important. There's interest in why the tau aggregates and it doesn't get cleared normally. So this might be failure of the lysosomal or autophagy pathways. In different tauopathies, different levels of this pathway seem to be involved. Like, so for example, in Alzheimer's disease, we don't know exactly how it works, but A beta is thought to increase the phosphorylation of tau by maybe activating certain kinases. For example, glycogen synthase kinase 3 beta, which is part of Wnt signaling, is one of the lead tau kinases that's been proposed. I mentioned before the frontotemporal dementia cases. They actually have mutations in the tau locus. There are others we call sporadic tauopathies, where these isoform levels that I mentioned get thrown off, and the tau protein, we'll talk a little bit more about this in a moment, has microtubule binding domain repeats, and alternative splicing can change the number of microtubule binding domain repeats. You can have either 3 or 4 repeats, and certain tauopathies will have increased 4R aggregates or 3R aggregates. So this is going to be important later as well. Others have like a mixed pattern where you see both of the isoform types accumulating. So this is a tauopathy safari, which is intended to show you all the different types of aggregates that can be formed. It's not complete, but you see tau accumulate in neurons. This is an example of a classic flame-shaped neurofibrillary tangle in a pyramidal neuron in the hippocampus that you see in Alzheimer's and other contexts. There are PIC bodies, which are little rounded ones. There are grains, little dots that appear in the neuropil. You can have granular cytoplasmic inclusions. There are rings, nuclear rings, so on. In glia, there are different types of astrocytes, tufted astrocytes, coil body, astrocytic plaques. One thing that's kind of interesting is that in these mutational tauopathies, you get these different morphological forms of tau. In the sporadic tauopathies, where there's not a mutation, you see the same types of forms accumulating, these tufted astrocytes and the grains and the PIC bodies and so on. So there's a lot of different tauopathies, and there's a lot of different signatures of them, and you can tell them by simply looking under the microscope. And if you get good at it and you train yourself, you'll be able to recognize these features in different brain regions and different symptoms, and then you can diagnose things. So this is where I became obsessed. So when I was doing my fellowship training, a number of cases came to autopsy with a clinical diagnosis of Alzheimer's disease. So they didn't have any of these other tauopathies with movement disorder or motor neuron disease. They were classic Alzheimer's disease clinically. They were certified Alzheimer's by some of the best neurologists in the world. But when they died, they had absolutely no amyloid plaques anywhere in the brain. And these are immunohistochemical stains from the frontal cortex and the hippocampal formation. The positive signal is going to be in brown. You're going to see a lot of these immunohistochemical stains in this presentation. So there's really no brown staining. There's no plaques in these cases whatsoever. compared to the Alzheimer's case here, where you see lots of these amyloid plaques. And even in normal aging, you see a lot of amyloid accumulation in plaques. So these cases really stood out. But what we did see, and this is what I'm showing here in panel E, the brown staining is, and this is a low power view, this is a hippocampus in the medial temporal lobe, lots and lots of tau accumulating. And that was the only neuropathological change that was evident in these brains. You can see a similar pattern in Alzheimer's disease with tau accumulating in the medial temporal lobe. You even see this sort of thing happening in normal aging. Usually people get neurofibrillary tangles in the hippocampus. But what really stood out about these tangle-only cases is that they had lots of tangles in the absence of amyloid in the medial temporal lobe, and they were demented, which is not a normal finding. So here are some pictures of some of these original cases that I was studying. This is an immunohistochemical stain for abnormal tau, and each one of these brown things is a neurofibrillary tangle. This is a classical silver stain. It's the same stain that Alzheimer used when he first described tangles in Alzheimer's disease. And you can see them forming these little filamentous aggregates. They're hair-like inside neurons. We even see lots of these tombstone tangles. These are the ghost tangles, the remnant of the tangle after the cell has died, and they're not quite as dark in these stains. These are more stains with antiserum that is specific to the different forms of tau. Here's a 3R tau stain, which is positive, and a 4R tau stain, which is positive. This is the same tau isoform profile that you see in Alzheimer's disease. And so the pattern, that matches. These are electron microscopic images of the tau filaments that we prepared in Alzheimer's disease and these tangle-only cases. And if you look closely, you'll see that you get these, it's a twisted paired helical filament, which is the classic electron microscopy signature. So these cases were developing Alzheimer's disease symptomatology kind of clinically. And when they died and we looked at their brain, they were developing Alzheimer's type neurofibrillary tangles that were regionally, morphologically, ultrastructurally, and biochemically identical to the tangles that you see in Alzheimer's disease. And so when I learned about these cases, I was hooked, right? If it's possible that people are getting Alzheimer's disease without any amyloid, that would be very, very meaningful. And the reason why is because it was thought that amyloid was the cause of Alzheimer's disease, and it still is thought to be the cause by many people. So how do we resolve this discrepancy? I wasn't the first person to notice this category. Actually, most pathologists who had looked at aging brains had noticed that there were a very small number of people, depend on the autopsy series, between 1% to 8%, that had this pattern. Each investigator or report kind of used a different name, so it was super confusing in the literature what people exactly were talking about, especially because the term tangle-only dementia can be confused with tauopathy in general. So if you see a tangle-only, are you talking about one of these other tauopathies? And it took a long time for us to kind of come to terms with what exactly were the features that we were talking about. But these cases were predominantly older, and when I say older, I mean oldest old, not young old. Regular old, 65 to 80. These patients were 80 to 100, so 90 years old was the average, and mostly women. The other thing that was kind of different is they were only moderately demented. They weren't as demented as the classic Alzheimer's disease that you think of. It's associated with plaques and ApoE and maybe mutations in APP or other genes. Also, they were localized to the medial temporal lobe. So one of the things that had been described in Alzheimer's is that tau progresses in a hierarchical manner through different brain regions, and it starts in the medial temporal lobe and then spreads to other brain regions. These patients did not spread. What they did get was very, very severe localized disease, as opposed to a spreading kind of wave through the brain. The hippocampus proper, the entorhinal cortex, the amygdala, these regions were where the tau was, but frontal, parietal, occipital, not that much. The parenchymal A beta deposits, not there. So what do you do with this? The reason why I'm so confusing is because Alzheimer's disease is thought of as the plaque and tangle disease. And it's funny because I thought that that's what it was, but it seems to be an opinion, and it depends on who you talk to. So most people think of Alzheimer's disease as plaques and tangles, and that's baked into the diagnostic criteria. So these patients that we were seeing, that I was confronted with at the microscope, essentially didn't read the textbook. So what do you do with somebody who doesn't have plaques and tangles? This is the current guidelines that we have for diagnosing Alzheimer's disease. It's a table that incorporates tangle staging, and we use something called the BRAC tangle staging that I was mentioning before. Early tangles in the medial temporal lobe that then get more severe and spread to the limbic regions, hippocampus, amygdala, and then spread diffusely throughout the neocortex. We also use a system for staging plaques. This is the thal phase here. There's another version of the plaque staging called CIRAD, where you take the regional distribution of your plaques, and then you come up with a probabilistic statement of the likelihood that this patient has Alzheimer's disease symptomatology or burden. The problem is these patients fall into this category: no amyloid, but lots of neurofibrillary tangles. And the criteria didn't give us any guidance about what to do with these patients, although there was kind of grumblings in the community. This is a study that was published by my close collaborator and friend, Pete Nelson, at the University of Kentucky, who worked with Bill Marksbury in about 2009. What is this showing? He grabbed a large data set with just plaques and tangle counts in the medial temporal lobe and the neocortical regions, and plotted them. And what you'll see here is in the neocortex, you have pretty good correlations between plaques and tangles. But if you look in the medial temporal lobe, what he's pointing out with this little blue arrow is that there's something different. There's a stack here of patients that are getting intermediate levels of neurofibrillary tangles, not as severe as you see when there are plaques, but they don't seem to be developing significant amounts of amyloid. So what is this group? It seems to be a different category, different trajectory. So I wanted to figure it out, and I didn't know exactly where to start. So we put a checklist together, and really the approach was to say, well, what do we know? What mechanisms exist that we know can cause tangles, and do they apply to these cases? And what a lot of people were telling me was that Alzheimer's is caused by amyloid, but they weren't just saying it was caused by the plaques. They were saying it was caused by a soluble form of amyloid that was invisible to the histological preparations. It's a biochemical change, and this oligomer, which is soluble, maybe is diffusing and messing with synapses and causing neurodegeneration in some way. Other people said to me, John, you just got to sequence the tau gene. You're going to find a mutation there. This is an FTLD, let's just move on. This is not so interesting. Other people, and they tended to be grant reviewers, said, this is normal. This is just a normal thing, and you're comparing normal to normal, and you're wasting my time and everybody's time. This is not an interesting project. But I knew that there was something different about them because they weren't normal. They were demented. So it was very hard for me to call it normal. But this idea that could have been some kind of pathological aging was intriguing, and we'll talk about that. I mentioned all these other mechanisms: viral, traumatic, toxic, something new. So these were ideas that were all kind of on the table. Let's start with the amyloid cascade hypothesis. So the amyloid cascade hypothesis has dominated our field for a long, long time. And the reason is because it's very compelling, and there's lots of evidence to support it. But basically, the amyloid cascade hypothesis states that A-beta toxicity from this peptide derived from the amyloid precursor protein is the cause of Alzheimer's disease. Everything else, neurodegeneration, tangles, whatever you're seeing in those brains, is secondary to this A-beta toxicity. And the evidence is very strong. So there are mutations in the amyloid precursor protein. Alison Goat discovered here, our leader in our Alzheimer's Center. There's these presenilin 1 and 2 mutations that affect the production of amyloid. There's the ApoE in sporadic Alzheimer's, which seems to influence deposition. It's very, very strongly correlated, that gene, with amyloid deposition. But then there's a whole host of biochemical, cell and animal models, data that's been published over many decades that all supports this genetic evidence. So how can you deny the genetic evidence? But there were smart people that I was meeting who disagreed. And what were their arguments? Well, the first argument is that A beta plaques correlate terribly with cognitive impairment. You have lots of people, lots of examples of high plaque burden with no cognitive impairment, or vice versa. For example, these tangle-only dementia cases that have Alzheimer's with very little and no plaques. The tangles correlate better. There are some forms of the amyloid plaque called neuritic plaques that maybe correlate a little bit better. There's the synapses, the synapse burden and atrophy is probably the best correlate. The proponents of A beta and the amyloid hypothesis have responses to all of this, of course. They say that, well, it's not the plaques that are important, it's this invisible soluble oligomer that you need to measure biochemically. In fact, they say the plaques might be a good thing. They're protective. They keep this horrible soluble toxic oligomer away from your synapses. It's like a sponge. They also were saying that, maybe the tangles are correlating, but A beta is the trigger, and that you need the A beta to come in and start the tangle to form, and then it disappears. It's kind of like a hit and run idea. And this idea of synapses is linking A beta to synapses. So there's another problem, a big problem. I call it the spatiotemporal disconnect. Tangles and plaques form in different parts of the brain at the beginning of the disease, and the tangles appear first. So we have neuritic plaques. So we have plaques, all plaques appear first in the neocortex. The tangles are appearing at the bottom of the brain in the medial temporal lobe. And brainstem actually can go back even further. The tangles are first. So if you have this tangles appearing first and a different region than the plaques, how are you going to say that those plaques are causing those tangles? So that's hard to reconcile. One of the possibilities is maybe those neurons in the medial temporal lobe have axons that project to places far away. You just need to map them better, and you'll find that their synaptic terminals touch a plaque far away in the neocortex. The other problem is that there have been several large phase 3 clinical trials targeting A beta, and have failed to show efficacy in the disease. And so this has caused a lot of anxiety in the field. So this is how we approached it. We started to think about the metabolism of A beta and where it comes from. And I just want to let everybody who's on the call know that this is an equal opportunity presentation. There's a little bit for everybody. We have some gels, but we also have some. Could you please mute yourself? Thank you. We have histology, and we have some cognitive stuff. And anyway, so just bear with me through these gels. Anyway, the point is that the A beta peptide comes from this precursor protein, and there are enzymes that cleave it. There's some enzymes called the alpha secretase and this pathway that cleave the precursor protein right in the middle. So it makes it so you can't make the A beta peptide. So this is called alpha secretase, and this is a secretory APP alpha fragment. There's another secretase, the beta secretase, that cleaves in a different spot, and that's followed by a gamma secretase, and that releases this toxic A beta peptide here. So when we went to measure the A beta levels in our brains, we did not find elevated soluble A beta. Instead, we found very low levels of A beta. In fact, the levels were so low, they were undetectable, lower than the levels of controls. And so that might explain why we have people who are in their 90s or 100 years old that have absolutely no plaques. And this is kind of interesting in and of itself. How do you make it to 100 and completely escape Alzheimer's disease? That's something we can talk about in a minute. We also measured the levels of these secretory fragments, and this non-amyloidogenic protective secretory APP alpha was highly overexpressed relative to the secretory APP beta that's the pro-amyloidogenic form. So we thought that this was compelling evidence that they have differences in the way they process their A beta and APP. Also, this ApoE allele that I alluded to before, which is so strongly correlated with Alzheimer's disease and amyloid deposition, was not correlated with tangle-only dementia in this first series of about 34 patients that we looked at. So we kind of concluded from this that this is not an A-beta dependent mechanism. Now, different people. One last thing I was going to mention. So this work was done by my very first postdoc, Ismael Santamaria, who's now an assistant professor, tenure track at Columbia University. I had an extremely talented master's student who worked with Soon-Ho Kim in my lab, Kofi Zorgby, who's now a medical student, MD PhD actually at Emory. And he came in and followed this up by measuring the activities of the alpha and beta secretase ADAM10 and BASE1 and looking at the ratios. And he built predictive models to try to tease out if the activity of these enzymes is influencing this. So of course, the ApoE allele frequencies is strongly correlated and a very strong predictor of amyloid deposition. But he was able to look at over 100 patients that had no or mild, moderate, or severe amyloid deposition. And using this cohort, he was able to show that the ratio of your ADAM10 to base is a strong predictor of amyloid plaque deposition. So anyway, the conclusion from this is not an A-beta disease. If you consider Alzheimer's disease to be the disease caused by amyloid, and you're a proponent of the amyloid hypothesis, then maybe you could argue that it is. But we think that it's not. And so we moved on to this idea of whether there was a mutation. So let me tell you quickly. This is a schematic showing the tau gene. We know there are mutations in the tau gene that cause frontotemporal dementia. Most of them are clustered around this exon 10. Alternative pre-mRNA splicing of exon 10 gives rise to these different forms with different numbers of microtubule binding domain repeats. So this is the first repeat, the second repeat, the third repeat, the fourth repeat, and you get forms with 3 or 4, or this is a problem with this figure, please ignore it. There are also alternatively spliced exons in the N terminus. Some of the mutations influence the splicing of exon 10, and some of them have amino acid changes that change the function of the protein. These mutations that change amino acids, some of them influence the binding of tau to microtubules, and some of them cause the protein to be more likely to aggregate. These other mutations that are near exon 10, they change the ratio of the different numbers of microtubule binding domain repeats. So we did next-gen sequencing. I put in Roche 454 here for people who are in the know, because it's kind of hilarious. It was the first gen of next-gen sequencing, but it was very fancy at the time. We sequenced the tau locus in these genes, and we could not find, using 454, we could not find any mutations in these diseases, and we could not find differences in the isoform ratios biochemically. We also couldn't find differences in the phosphorylation pattern, these other structural changes. So we don't really think this is an FTLD with a tau mutation. We just crossed that out. But what about this idea of like normal aging? So the aging literature is really kooky, but the idea that it's like normal, I don't really want to go there, but I'm just going to point out that they're demented, and that this dementia is strongly correlated with cognitive impairment. In order to do these studies, we leveraged the NAC neuropathology data set. The National Alzheimer's Coordinating Center, the University of Washington, aggregates data from all the Alzheimer's centers. It's extremely useful data set, and if you want to do studies in the neuropathology of AD, you can leverage it and everybody can get access for free. And I teamed up with some of the folks there at the NAC who also shared my interest in part, and we pulled out 377 patients that had matched clinical data, and were able to show a lot with them. And we had actually, it was a highly productive collaboration. We published 5 papers together looking at the clinical pathological correlations in part. The first paper was led by Lila Besser, and she showed beautifully that cognitive impairment in these amyloid negative subjects is associated with the stage, the BRAC stage that I was telling you about. She was also in a follow-up study where she looked at different cognitive domains, a global composite, episodic memory, attention, executive function, semantic memory, that there are differences between these amyloid negative and amyloid positive patients. And specifically, there was a sparing of semantic memory versus Alzheimer's disease in part. we worked with another member of that team, Marilee Thielen, who did the she did the longitudinal studies, and she was able to show that across the board, over time, patients that had a diagnosis of Alzheimer's disease or MCI, cognitive impairment, who then came to autopsy and found to have PART, were very different clinically. Their clinical trajectories were very different than the classic plaque and tangle ADs. Across the board, really, we looked at the same domains as well as the CDR sum of boxes, and with much more severe malignant disease if you have plaques, which is not surprising. And that also goes along with the original reports of these tangle-only cases. What about pathological aging, path aging? So you may hear this term in the literature get kind of thrown around. What is pathological aging? Well, I mentioned this before, the aging literature is a little bit kooky. So for some reason, it attracts certain type of person with certain ideas, but there are some really, really good scientists out there who are working on aging. And one of the groups, this Rowe and Kahn study that was done in 1987, set up this model that I think is really attractive. What they said was, the way that we define normal is kind of wrong. Most studies will say normal is the absence of meeting the criteria for a specific disease. So if you don't meet the criteria for Alzheimer's disease, you don't have Alzheimer's disease, and then you're in the controls, right? But if you look at those controls very carefully, they often have a lot of pathology. And that's where the interesting stuff comes in. So they said, look at your normals, and you'll see that some of those normals are functioning about as well as their age-matched kind of their peers. There's another group of normals that are functioning as well as their younger counterparts. So if you're 90 years old and you have memory problems, you're going to be in the majority, because most people in their 90s will have memory issues, age-related memory impairment, subjective memory complaint, what have you. But if you compare them, if you have a 90-year-old with a perfect memory that's just as good as a 20-year-old, that really stands out. So conceptually, they define these categories of successful aging versus usual aging. And that can happen in any domain. It doesn't have, you don't have to think about just the brain, but any organ, you can have a successful cerebral aging, but you can also have a successful aging of your liver or your kidney, or what have you. So I really was intrigued by this model, and I was wondering if we can kind of implement it in our study. So we started to think about tangle-only dementia as just the tip of the iceberg, and then tangle-only mild cognitive impairment and tangle-only kind of normal memory. And we, instead of looking at the BRAC hierarchical progression, because these patients their tangles don't spread from the medial temporal lobe, we had to use different measures of tau burden. One of the key things, and this is the first bullet here on the slide, and I'm going to tell it to you last, is that tangles are ubiquitous. Everybody gets them. It's just how many you get. I showed you that at the beginning in one of the earlier slides. It's just the number goes up as you age. By age 50, everybody's got tangles in their hippocampal formation. In your teens, people have tangles in the brainstem. So you can't think of this as case control. You have to think of it as a quantitative trait, and you have to redesign your studies. And when we started thinking in this way, then some really interesting genetics and other things started to come up. And this is the first thing that we noticed. So the tau gene is in an unstable genomic locus, and it's a real slog. There are lots of duplications and repeats and rearrangements, inversions, duplications. Every single way that a gene can be scrambled, it's happened in the tau locus. If there's anybody here who's a Drosophila researcher, this is like a balancer chromosome that's just so messed up, it can never recombine with the regular form. And we have these two versions, we call them the haplotypes H1 and H2. The H1 is probably the ancestral version. H2 has been maybe proposed to come from Neanderthals. We don't really know. And here the allele frequencies are being shown. But this H1 haplotype is strongly associated with tauopathy versus the H2, which seems to be protective. It's also associated with Alzheimer's disease, but the effect size is much smaller. So when we stratified our aging cases by successful aging, control, and these tangle onlys, we started to see this trend. The successful agers had high allele frequency of this protective H2, whereas the usual agers were in the middle, and the tangle only cases had an enrichment for the H1. So we were started to think that these cases have a different genetic risk profile, and that was one of the drivers for doing more genetics later on. So we're really into this idea of a kind of pathological aging. Now, what about chronic traumatic encephalopathy? So you may have read about chronic traumatic encephalopathy because it seems to be in the news like twice a day, or it had been. Maybe it's now like once a week. It's such an important issue. Unlike Alzheimer's disease, this is a disease that affects young people. So the questions around chronic traumatic encephalopathy, which occurs in traumatic brain injury, repetitive traumatic brain injury in athletes, is important. Should children be allowed to or encouraged to participate in contact sports or combat sports? It's super important for the military, blast injury, military veterans, domestic violence. So this CTE is a long-term neuropathological change, a degenerative change that happens months or even years after this exposure. There's a lot of interest from a lot of different domains. So are these cases we're talking about CTE? Well, one of the things that's different is CTE tends to happen in athletes and football players, and most of them are men, at least in the autopsy studies that we have. These tend to be 90-year-old women in this category, although there is overlap and there's differences. And so the risk is not exactly the same. I was fortunate enough to get involved with a group of neuropathologists who were building consensus criteria for CTE. So I had an opportunity to study a large number of cases. And everybody should know that we have expanded dramatically the number of cases in the collection in the brain bank with not just chronic traumatic encephalopathy, but also moderate and severe TBI through collaborations with Kristen Dams-O'Connor, who's here at Mount Sinai. And I just want to point out, that's one of the other amazing things about Mount Sinai is that you'll find outstanding experts in pretty much every aspect of brain disease or normal function. So it's super synergistic. Anyway, what am I going to tell you? I'm going to tell you about the signatures of CTE and how they differ from this tangle-only pattern. So CTE is a biomechanical trigger. Forces get applied to the skull, forces get transmitted to the brain parenchyma. It's kind of a geometry problem, the way the structure of the brain, the biomechanical properties of the tissue, the elasticity, the forces are applied, there's stretching, shearing, compression that then damages neuronal elements. And because of that, you see specific parts of the brain that are biomechanically vulnerable where the tau appears. So this is an example of a sulcal lesion. If you think of the brain as a Kit Kat bar, you can think of the gyri and the sulci of the different, each of the different bars of the Kit Kat. When you break a Kit Kat, it doesn't break at the thick part, it breaks at the depth of the Kit Kat's sulcus. And so this is what we're seeing here, is that the forces are being transmitted, and all these little dots here are cells with neurofibrillary tangles in them. You also see tangles around blood vessels in CTE. This is a small arteriole, and these are all neurons and glia surrounding it. And that's because the arteriole has a rigidity that's over 100 times higher than the soft brain parenchyma around it. And so you imagine the brain is a bowl of jello. If you bake in a rigid tube and you shake that bowl of jello, all the shearing and forces are going to be applied to that interface between that blood vessel and the parenchyma around it. So we can find this lesion, and when we see it, it's diagnostic of CTE. Now, when we looked at our cases, we didn't see these neocortical sulcal lesions in the tangle-only cases. We don't see the perivascular tau. The thing that does overlap, however, is the medial temporal lobe predominance. And so in CTE, this is a 57-year-old NFL player with a severe atrophy of the hippocampal formation in tau. It's the same distribution as this 94-year-old woman here with high tau deposition in the medial temporal lobe. Now, the pattern is not the same, but it does overlap. And when I started thinking about this, and I worked with a very talented, brilliant neuropathologist at Columbia University named Jean-Paul Van Sottel, and he trained with E.P. Richardson at Mass General. And the two of them were always interested in CTE and why it causes tangles. And they had come up with a hypothesis, but never published it, but it was passed down to me through Dr. Van Sottel. The hypothesis was that maybe tangles are caused by microtrauma, because they always saw tangles in certain parts of the brain that they thought might be vulnerable to trauma, particularly the anterior part of the temporal lobe. And they said, well, maybe there's something called, like, not mild TBI, but maybe very mild TBI, a microtrauma that accumulates over your lifetime. And I've always liked that hypothesis. And if anybody wants to collaborate, we can work on this. But if you look at the CT, you look at the tangle-only cases, I'd like to point out, this is entorhinal cortex in the medial temporal lobe is a pressure point. It's where the tentorium of the cerebellum interfaces with the cortex here. And we see that we have humans have evolved these large brains and these large frontal cortexes, and it's changed the geometry, put the hippocampus at the bottom. So there's this geometry. There's a bunch of reasons. I'm going to tell you the 3 reasons why I think that this is a reasonable hypothesis. Is that the geometry matches, there's overlapping regional vulnerability, and actually, if you look at brains of people, there's a notching at this precise spot where the tangles first start, which is physical evidence of the tentorium pushing into the brain. And so if you pull out a bunch of CTE cases and you look at that notching, that uncle notch, and you stain for this abnormal tau, you can see that's where it's developing early in cases. And so this needs to be followed up on, but I think it's intriguing. Now, after all of this, we had convinced enough neuropathologists that these cases with this tangle-only pattern were somehow different from classic plaque and tangle Alzheimer's disease. They were different enough that they deserved a proper designation. And so we worked with John Trojanowski, Julie Schneider, and a lot of other people at ADRCs and brain banks across the world to hash out some criteria and give it a name. And we came up with the name primary age-related tauopathy. Pete Nelson at the University of Kentucky, who I presented some of his data early in the talk, was highly involved in these discussions as well. What started as a review article became these consensus criteria. I'm thrilled about it because people have started to use this designation in research, and it started to pop up in many autopsy studies of Alzheimer's disease. These are the criteria here. It requires the presence of neurofibrillary tangles in the medial temporal lobe, so that's Braak stage 4 or less for the connoisseurs of tau staging. Absence of any other tauopathy. You cannot have significant amounts of A beta. How you define a significant amount of A beta is up for debate. There were different categories, a definite part or possible part. You didn't have to do any of these other studies, but you could do them if you want. Now, one of the things that was like most surprising to me is that there was a lot of pushback. And I thought that the amyloid community would be really, really upset. But in fact, they were not. It was the tau community. And there's a group of Europeans, Heiko Braak himself actually, who believes that these part cases actually represent Alzheimer's disease, just a very early phase of Alzheimer's disease. And that had they lived longer, they would have developed plaques. In fact, the plaques are caused by the tangles. And so they've really flipped the way that and I think it's fascinating, flipped the way we think about Alzheimer's disease on its head. If tangles come first and the plaques are coming later, as I mentioned before, then, you know, tangles should cause the plaques. Now, I've pulled this figure from Brack's paper. These are his autopsy series results showing the Brack stage over the aging spectrum, and you'll see that the earliest Brack stages come in here in the teen years with his tangles in the brainstem, and they progressively get more and more severe, and that the amyloid plaques come in later and they become more severe as you progress. One of the things that they're not showing here is that over 100, the plaques start to come down, and there seems to be a wave of amyloid that comes and goes, and that the tau continues to progress. We need more longitudinal studies to kind of sort this out, but it is controversial. Why is it useful? Well, it's been informative for biomarker studies. There's a lot more interest in trying to diagnose Alzheimer's disease and other neurodegenerative diseases while people are still alive. The definitive gold standard diagnosis remains the autopsy. And it would be much easier to do a clinical trial if you knew what people's diagnosis was before you enrolled them. So we're finding that a good 30% of patients that had Alzheimer's disease clinically, when they do these amyloid PET, CSF, A beta stains, or A beta measures, they don't have amyloid pathology, and so that's disconcerting. The biomarker teams have come up with a category they call suspected non-amyloid pathophysiology, and these are people that have tau with these biomarkers measures, but they don't have any amyloid. And so the main point is that tauopathy in the population is very, very common without A beta amyloidosis. Another thing that's been useful is to kind of understand the spread of tau. If you're in the field, you may have heard about this prion-like propagation of tau, and there's a lot of research that's gone into that. A lot of the ideas have emerged from Stanley Prusner's work at UCSF. These cases have shown that by studying these populations, this is with Lila Besser and the NAC group that I mentioned before, when we looked at cases across the Brack distribution, the plaque negative cases almost never get up to these higher neocortical stages of 5 and 6. As soon as you start to see some neuritic plaques, the 5 and 6 levels surge. So from looking at this graph, and others see the same, interpret the data in the same way, is that A beta is the kind of neocortical spreader. The idea is that as soon as you have A beta, it allows the tau now to kind of spread out of the medial temporal lobe. An alternative interpretation, which I tend to favor, is that these are actually two different trajectories. They're two different diseases or two different categories. Another thing that we're able to do is we're able to launch a larger study. And the goal of this study, which we call the PART working group, was to develop neuropathological staging systems that kind of go beyond the Brack stage, because this hierarchy is not as important when you're looking, the neuroanatomical hierarchy is not important as when you're looking at burden in a single region. So we're trying to do this better. I'm not going to show any data, but we're spending a lot of time trying to use computer-assisted morphometrics and machine learning and deep learning to stage PART and other neurodegenerative diseases later. The next time I'm invited to present at the Cyber Series, we'll have a lot to show. But we're getting started and we're building this large cohort. We teamed up with Chuck White, a neuropathologist and director of the ADRC neuropathology team at UT Southwestern, and his trainee, Jamie Walker, who's now got her own group in Texas as well. And Kurt Farrell, extremely talented postdoc and now recently promoted in my lab, who led this effort. And the goal was to develop these staging strategies, but then to use them. And we use them for clinical pathological correlations, but also biomarker and genetic studies. And really the hope is that these genetic studies, along with the histology, will pave the way towards mechanistic discovery and then give us a shot at some rationally designed therapeutics for PART, but also hopefully they'll work for other tauopathies. That's what we're doing. If you think about it, this is translational research going in the wrong direction. People want us to go from the lab to the bedside. We're kind of going the wrong way. And it's probably going to take us a lot longer, but we think we're going to be able to move from our patients to the lab, model disease, and then come back to the patients sometime down the road. One of the first things that we noticed, we collected close to, well, 914 hippocampi from ADRC brain banks across the nation, and started to compare their tau distribution. One of the things that came out first, and Jamie published this just this year in JNEN, is that PART cases have a severe and early vulnerability of the cornuamonas 2 region of the hippocampus. And so this is, you can see this right here. This is the human hippocampus. This is CA1. This is the subiculum. This is the dentate gyrus. And you can see this deep and dark scar in this AT8 phospho-tau stain. That's very different from regular Alzheimer's disease, where CA2 tends to be spared, as is other regions, but you have a lot of staining, tram track staining here in CA1, subiculum, entorhinal cortex, and there are a couple of examples here. Another thing that we were able to do with this large study, and this was something that Kurt did with Megan Ida, another extremely talented trainee in the lab who's now a medical student at the University of Michigan, was they took the data set and compared the PART distribution to chronic traumatic encephalopathy. So this is actually a really, really important question from a medical legal point of view. If somebody develops a dementia and they played in a contact sport, people want to know, did that dementia come from CTE, or is this just normal aging? And don't listen to the NFL if they tell you that it's all just normal tangles, because it's not. And one of the things that she was able to show, and this, by the way, was a pandemic project, and was enabled by lots of digital scans of hippocampi that were then put on a hard drive and taken home and segmented and quantified. What Megan did was she took a group of age-matched PART and CTE cases, segmented the hippocampus, and then compared the tau burden using periopositive pixel quants. And what she found was that in CTE, there was a marked increase in the relative proportion of tangles in the CA4 region versus PART, which was generally protected. She also saw increases in other regions, but not in CA2. And we had said that CA2 seemed to be infected early in PART. It also is regions affected in CTE. And so this is potentially a way that we're going to be able to use this knowledge to differentiate them. So what about the genetics? So I'm going to cruise through this last thing here quickly. So Kurt was building this large cohort so he could use the tau burden as an endophenotype. And we collaborated very closely with Alison Goat to do this genetic study. We had input from lots of other people. But we can't thank Alison enough for all the support that she gave me and Kurt in doing this. Kurt used Braak stage as a quantitative trait and did a GWAS. And one of the questions was, are the genes that are important for Alzheimer's disease associated with PART burden, but also are there other genes? And so one of the things that came out of the genome-wide study was a locus on chromosome 4. Here's the SNP. They contain 3 genes, JADE1, CORF33, and SELT1, that reach genome-wide significant in this cohort. And this is our Manhattan plot showing that we're just crossing the threshold here. This is the locus zoom showing the JADE1 gene here. And there are 3 genes total. Now, what's important to recognize is that these SNPs are marking a locus, and they don't tell you which gene is important. There are other ways to use RNA expression, these EQTLs. When we started to use EQTLs to try to map these, we didn't have much success. We're still working on that. But we're focusing on JADE1 for the reasons I'm about to show you from some of the data that we collected. And we used other orthogonal approaches to try to validate these hits. So JADE1 is a gene with very little known function. It's not well studied. It is a member of a family of 3 proteins, JADE1, 2, and 3. It stands for gene for apoptosis and differentiation. I don't know how they flipped the J for the G, but it's cute, so it helps you remember it. Functions include histone acetylation, cell cycle. This is a von Hippel-Lindau tumor suppressor gene, which is associated with certain vascular tumors. This is one of the first known binding proteins of JADE1. It was identified in a screen of apoptosis, which is interesting and important for neurodegenerative diseases, and also Wnt signaling. It's an inhibitor of Wnt signaling, which is really interesting because GSK3 beta, which I mentioned before as one of the lead tau kinase candidates, is an integral part of Wnt signaling. And so one of the more powerful pieces of data that we collected were these co-IP experiments. So Soon Ho Kim in the lab took human part cases and other tauopathies and asked the question of whether or not there's a direct physical interaction between JADE1 and the tau protein. And when he pulled the JADE1 out in an IP and then probed those samples for tau, he found that there was co-aggregation or co-immunoprecipitation of tau with JADE1. And this interaction was interesting because there's all these different tau isoforms that I mentioned. There are 6 of them, prominent ones in the brain. And this is just a control here. He only saw a band that was about the same size as this 0N4R. So none of those N-terminal inserts. It just had 4R exon. When he did it the other way around and he pulled out the tau protein, he found that this JADE1 also came along for the ride. And you probably wondering what this S is. There are 2 different isoforms of JADE1. There's a JADE1L and a JADE1S, a long and a short form, and they probably have different functions. These 2 bands here, by the way, in the control, the input and the IP have different sizes, and it's because JADE1 gets phosphorylated and it loses its phosphorylation, which we showed here after the IP. And so we think these are the same band, even though they're migrating slightly different sizes. Another weird thing, so we confirmed that this 4R was specific. So here we're probing the IP blot with a 4R tau specific antibody, and we're seeing something there. When we do the 3R antibody, we don't see anything. And this tau that's coming along with JADE1 is abnormally hyperphosphorylated, and we have a whole bunch of different antisera that can detect those forms, the abnormal hyperphosphorylated forms of tau, and we're seeing them in all of them. So this was really interesting and different, the possibility that JADE1 was specifically aggregating with 4R tau. I didn't emphasize this earlier on in the talk, but 4R tau is thought to be particularly toxic, more potent toxic than other forms. Anyway, so Kurt did an immunohistochemical study of many of the tauopathy cases that we're interested in, not all of them, where he basically took that antibody and asked whether or not JADE1 was present in the tangles. So did it localize to these abnormal cells that are dying with tangles in them? And he found these are stains showing the tangles here. So the same field, we see tangles. I'm highlighting them with my little arrow here. All these little dots are JADE1 positive tangles. If you look at Alzheimer's disease, you see the same thing. You also see around the amyloid plaque, you see JADE1 positive positivities. These are degenerating neurites around the amyloid plaque. The chronic traumatic encephalopathy cases, you're also kind of seeing it too, and as well as progressive supranuclear palsy, which is a specific 4R tauopathy. Part AD, CTE, these are mixed tauopathies, meaning 3R and 4R. And this 4R tauopathy, you see it. What really blew us away, we've looked at 3 Pick's disease cases, which is the 1-3R tauopathy, and none of those tangles, none of those cases were positive. So this goes along with our data that tau, only 4R tau is aggregating with JADE1 and not the 3R tau. So what do we have here? Is it a Bobby Dazzler? So this is intended for the small minority of people. I'm opening up the view here. I want to see if anybody's smiling except for me. So I know,
you know, John, it is 2 o'clock now, so you should wrap up soon, okay?
So that's it. That's where we are. So is part the exception that proves the rule, or is part of Alzheimer's disease? The jury's still out. And these are all the people whose ideas I stole, especially Kurt and Soon and Allison and the whole team. So thank you so much.
Wonderful. Thank you, John. So unshare, please. And then even though we're a minute over, let's give time if anyone has a burning question to ask. Very nice talk, John.
Thanks, Eric.
Any
You know where to find me.
Yeah, absolutely. Any questions?
Dr. Crary, I have a quick question.
Go ahead, Sarah.
One of the things that you emphasized at the beginning that I thought was interesting was the actual lack of amyloid in your PART patients, where they had less amyloid than controls. And I'd be interested to hear your thoughts on that difference and if that's something that you've looked at all.
Yeah, so why do they have less amyloid than controls? So yeah, there's two things that are interesting about them. Why do they get tangles? And probably maybe the more interesting question is why don't they have plaques? To make it to 100 years old and to have no plaques, that's successful plaque aging. They have a different genetic profile. We see for sure that they have a high allele frequency of the ApoE epsilon 2 allele, which is the opposite of E4. There are other alleles that Kurt has picked out that we think are important. And we think that they're influencing base and alpha secretase activity, and it needs more work. You can ask Kurt that.
Excellent. So we should move on since it is after 2. John, thanks so much for a great talk. You know, it emphasizes to me the complexity of dementing illness and the importance of establishing more of a pathophysiological understanding of different variants of dementia, and the work that you've described is really elegant and beautiful. Thank you very, very much.
Thank you for having me.
Yeah. Thank you, everybody. Thanks for joining us.